Right. Hello and welcome to the 2021 SCID Compass Summit. My name is Alyssa Creamer and I'm the Director of Specialized Projects for the Immune Deficiency Foundation. Here today are Michele Caggana and Colleen Stevens, both of the New York State Newborn Screening Program. They will be presenting on 10+ years of screening for SCID in New York, an evolution of primary and secondary molecular screening. If interested, a PDF of their slides can be found and downloaded on the session's main page on the virtual platform agenda. A recording of this session will be uploaded to the virtual platform shortly after the session ends. Please welcome Michele and Colleen.
Hi, everyone. I want to thank the Immune Deficiency Foundation for inviting Colleen and I to speak today, and thanks to the many people behind the scenes who helped support this virtual meeting. Today, I'm going to review for you the experience in New York State after 10 years of screening for SCID, and Colleen will follow with updates on our most recent sequencing work. Before I begin, I want to thank Denise K for compiling all the data that I'm going to show you this afternoon. Okay, it's not moving on this slide. Okay, there we go. Okay, so first we're going to start off with a bit of history. So SCID was first nominated for the RUSP in 2007, and of interest, it was the first one to undergo formal evidence review. This review was completed in 2009, and the recommendation was made not to add it because a baby had not yet been found by newborn screening, even though 2 states, Massachusetts and Wisconsin, were screening at the time. So then it was nominated again in 2010 in January after a case had been identified in Wisconsin via newborn screening. There was discussion and then it was voted upon at the same meeting, and the recommendation was sent to the Secretary of Health and Human Services. And the recommendation was ultimately approved in May of 2010. This was kind of important because SCID was the first newborn screening test that actually had a molecular component, although it was not genomic DNA. As you know, we're screening for a byproduct of T-cell receptor maturation. So today I'm going to discuss for you the New York evolution of SCID screening. And suffice it to say, though, at the time, the community was very happy when in 2018, all the states were screening for SCID. And even this map doesn't show Pennsylvania, but because newborn screening is done by contract in the state was screening, babies were being screened for SCID, even though it was not formally on the Pennsylvania panel at that time. One thing I'd like you to appreciate is this was a big lift for programs because there's many challenges with implementation of SCID. Considerations included space and arrangement of workflows, because this is a little bit different. There are different requirements for molecular testing. When we're looking for the absence of a marker, special challenges are presented because we're amplifying amino acid and we want nucleic acids, and we want to avoid having a false negative result. Programs also had to purchase new equipment, and in some cases, new staff had to be onboarded. So the countdown to SCID is what was going on here in New York State. We've been working on the development of an automated assay, which began in December of 2009. We were made aware of NIH funding, and the goal of this funding was actually to expand the number of babies who are screened for SCID in the United States. Dr. Ken Pass, who was the previous director of the newborn screening program, began work on this application, and then this work was transferred to me after he retired. The study that we oversaw included not only New York, but also screening in the state of California and Louisiana via the Wisconsin newborn screening program, and also screening in Puerto Rico via Massachusetts. All told, the project screened more than 800,000 babies, and we validated the assay prior to this and submitted it to the Clinical Laboratory Evaluation Program in New York, which is a required regulatory step for us. We also had to submit an emergency regulation, and that the CLEP and the emergency regulation was both approved on September 27th, 2010. So SCID screening began in New York State on September 29th, 2010, and I always remember that because it was my 20th wedding anniversary. Our first SCID baby was detected in New York in December of 2010. And the regulation to add SCID to the New York State panel was finally amended and approved in April of 2011. We ended up also receiving financial assistance via funding from the Model Foundation to continue screening beyond the NIH study period. So I'm going to talk a little bit more about this presumptive positive category in the next couple slides. We put through the emergency regulation, but the commissioner officially added SCID to the panel, and everything got approved in April of 2011. So let's start with a few definitions. Some states use the term presumptive positive to denote that the baby is being sent for evaluation. We use the term referral, and for our algorithm right now, that is less than 125 TRECs. We use the term presumptive positive as or borderline result, and these are babies who have TRECs between 125 and 200 TRECs. And in these cases, we request a repeat specimen for the baby. We also have another term for our algorithm called the repeat pre, and that stands for preterm infants who are born at less than 37 weeks gestational age. And those babies have TREC values less than 200. We ask for a repeat specimen from those babies once they reach 37 weeks gestational age, and then they're referred if they meet the criteria for referral. However, if we have babies that score a 0 TREC, even regardless of their gestational age, those babies get referred. So I'm going to walk you a bit through the evolution of our assay. We started out very conservatively, and our providers, we have 11 specialty care centers in New York, and they wanted a referral for any baby who had low TRECs. I remember the conversation. And as you can see on this slide, they got somewhat bombarded using the conservative cutoff of 200 in our state. And at that time when we first began, we had no borderline category. So any baby with TRECs less than 200 were referred for flow cytometry. So that only lasted a few months, and we looked at some data. And then in January of 2011, again, we did two things. We reduced the cutoff for referral at a TREC less than 150, and we also added the presumptive positive or borderline category. So any baby at this point with a TREC value between 150 and 200, a repeat sample was requested. And that dropped the referral rate somewhat and became a little bit more tolerable. We continued screening using this algorithm for a few months. And then up until 2014, we settled on the algorithm shown on this slide. So we looked at the data and we were able to reduce the initial cutoff to less than 125 TRECs. And the PP category basically used that same criteria for the cutoff as 125 to 200. The number of babies referred stayed about the same per month, but the SCID center directors were all okay with managing that volume of babies being sent to them. So we ended up being told that we had to actually move, and that was in 2014. And in doing so, we had made some revisions to the assay. We kept the cutoff the same, but we made some internal changes. And those changes actually resulted in improved screening. So now the number of babies that were referred were only about 8 per month. And that was a much better way to handle the babies were being referred and the smaller number were being referred. And so that lasted for about 2.5 more years. So what did we learn from all of these changes? Addition of the borderline category and various assay tweaks that we did over time reduced the number of referrals or the false positives by 87%. So that was a lot, obviously. And we were impacting far fewer families overall because we weren't asking for repeat heel sticks as frequently. And then we also were reducing the number of babies that needed evaluation. The positive predictive value, which is the number of babies who screen, the probability that a baby screens positive and actually has a diagnosis in the end, also improved. So it went from 0.6 to about 5.1%, which meant that about 1 in every 20 referred babies ended up having a confirmed diagnosis. We did make that change for the 0 rule for premature infants because premature babies can have SCID also. And we actually did have one child in that category who was born premature and ended up having SCID. So again, regardless of the baby's gestational age, if there's triple zeros, we call them, no TREX detected, we obviously refer those kids. At the time, we were also wondering if we could resolve some of our outcome data using molecular. So stay tuned for Colleen's talk after this. So as you might have noticed at the bottom of the last algorithm slide, there was a little comment in the left. And at the time, we were also preparing to add spinal muscular atrophy to our panel, along with 2 additional conditions. And this was scheduled for October of 2018. So up until that point, we had used that algorithm I showed you. SMA screening is also a molecular assay, and this one looks at genomic DNA for a specific deletion in the SMN1 gene. The beauty of this is that you can multiplex it with a SCID assay. And so uptake, the path was already in place for molecular testing in newborn screening laboratories with SCID. The advisory committee reviewed data and sent a recommendation to add SMA screening to the RUSP in February of 2018, and Secretary Azar approved it in July of 2018. We had already been working on a pilot study with consent looking at SMA and decided to add that to our universal screening panel because it was easily multiplexible, and we were adding mucopolysaccharidosis type 1 and guanidinoacetate acetate methyltransferase deficiency at the same time. So we ended up accumulating data, and as this slide shows you here with the black bars on the top, when you look at the old assay and then you look down, you can see to the left that the SCID, the SCID quantification actually shifts to the left, which means that the TREX are actually amplifying better. And so what that means is that if you have a baby who maybe in the old assay screened at a 180, and then would be followed up for a repeat screen, in the new assay, the baby might actually be screening at a 350 or something like that. And so that baby would be screen negative. And so we were concerned that we might miss a baby and have a false negative. So the changes to the assay, like I talked about when I moved, but they resulted in this sort of odd shift. And so we had to rethink what we were doing again. So we quickly assessed the data and changed the algorithm to what is shown on this slide. So what we decided to do, what we decided to do was any baby who had TREX less than or equal to 300 on that initial screen, using the multiplex assay, we then broke out and we used the old TREX assay to do what we call the retest. And that retest determines whether or not that baby goes for either diagnostic evaluation or we end up having a repeat screen. And so by making this change, we were able to basically sort of report out like the old assay, but do the new multiplex assay. And again, the changes were all internal, so externally, as far as our providers were concerned, nothing really changed. And it was interesting because when we did this, we also had a couple babies who we caught using this assay who ended up being diagnosed ultimately with Omen Syndrome. So this table shows the categories for confirmed SCID screening. And what we did was, these are codes that we use in the computer, and you can appreciate that there's many different types of, many different causes of SCID, and people are well aware of this. We wanted to track sort of the molecular cause by the genetic cause and also the phenotype. And so we have here a list of the codes that we use to really break down the different categories of SCID. And these are also taken from an expert guideline by the Clinical Laboratory Standards Institute, and the reference is shown there on the bottom. The next set of codes that we use in the system are those that define leaky SCID and other conditions that are associated with immunodeficiency, such as Charge syndrome, Down syndrome, and DeGeorge. And we know we detect some of the infants with these other conditions, but we know we don't detect all of the infants with these conditions, but they're all associated with some form of immunodeficiency and are positive on our assay. And this table shows additional ways that we categorize infants after evaluation. Some of these babies have low T cells at birth, may have birth defects or unknown causes of their immunodeficiency. We also show variant SCID on this slide, and these we count as confirmed cases because these kids have T cell counts that persist, but we don't know the genetic cause of that condition. And then lastly, we have codes that include our false positive results, the prematurity causes, and idiopathic T cell lymphopenia as a cause of low TREX. And then we also have to categorize other things like if the baby has passed away or the family was lost to follow-up. So when we look at the whole program from its start in 2010 through December 31st of 2020, we've screened almost 2.4 million babies. And of those, we've referred 1,664 for diagnostic evaluation, which is about 7 births per 100,000 screened, or 10,000 screened. So pretty good overall stats. When we look at some of the data graphically, this slide will show you the number of referrals by year through the end of 2020. The value to the left of 2010 is low because we started screening towards the end of that year. But you can appreciate those high number of referrals that I talked about that are related to the different algorithms that we started off with between 2011 and 2013. When we moved to the new lab and the assay was improved substantially, you can see a significant drop in the referrals. And so it's kind of interesting how you can, track the referral rate with changes that were made to the assay over time. In 2018 onward, you see a little bit of an increase, and these were the changes that were associated with that, changing that to that -300 cutoff with the multiplex with SMA. When we look at the diagnoses on the bottom of the slide, you see the tiny green bar. Those are the kids that were diagnosed ultimately with SCID, leaky SCID, and variant SCID. And you can appreciate that that number is fairly low, but fairly constant across all the years of screening. This graph also shows the real high false positive rate in the early years, and that's the red bar. So all the babies that were either referred or referred over time that were found not to have SCID. And then you can appreciate how those changes impacted that rate by looking at it in this graphical way. And it appears that we're also detecting maybe some additional forms of immunodeficiency shown by the blue. And that seems to have increased when we changed the assay when we added the SMA screening multiplex. And so having that 300 cutoff may have helped us kind of capture some of those kids that might have otherwise been undetected previously, even though the rate of SCID remains constant. This shows a breakdown of the genes, the different cases that I showed you on that other panel that had the various genetic causes and the leaky and variant SCID. So you can appreciate we still have a pretty large majority where the gene actually wasn't specified yet, and this maybe because the provider didn't actually send the sample out for molecular screening. This shows the cases of variant SCID by year, and note it's a very small number. There's only 12 of them overall, but these babies have very low TREX at birth with a low average of 56 TREX on the initial screen. So that's a fairly low number for us. And variant SCID is classified by us as lymphopenia with functional T cell impairment, but the cause is unknown. And these kids are deemed at higher risk for infection because of the low T cell count, even though they have normal mitogens. And this is a picture of all of those non-SCID cases. So again, these are other syndromes of T cell impairment. George, we detect some, like I said, we know we don't detect all of them in the population here, and then secondary T cell, and then a very small number of B cell deficiencies. So this slide actually shows you a really important aspect of SCID screening where we can actually learn new things. So we detect a pretty sizable population of babies who have this outcome called idiopathic T cell lymphopenia. And idiopathic just really means we don't know the cause. And these babies that are shown on this slide are those that have a diagnosis after the retest on the multiplex. And these babies overall have very low TREX, again, with a mean of about 63, and the median is 63 as well. So it looks like we're actually capturing more of those babies since we initiated the multiplex assay again. In reality, we capture two groups of babies. We capture the kids who have persistent T-cell lymphopenia, which are the ones that are shown on this slide. And then we have a whole other category of children who are originally diagnosed with this because of low T-cells when they're assessed, but then the T-cells resolve and the counts improve over time. But right now, we can't figure out which path the baby will take early on, and there's no real standard of care for these children with respect to vaccinations and overall management. So many of these kids are being followed in the centers over time, and we have a project to look at maybe we can identify some possible genetic determinants. We don't have a whole lot of these cases, but more to come in the future because this is a project that we're looking at to see if we can make any predictions about which path a child would take when they have this diagnosis after screening. So what about all the other referrals? About 1,000 out of the babies that we referred to the specialty care center over the entire time of screening were closed as no disease for a variety of different causes. And with regard to the idiopathic T-cell lymphopenia kids, 20 of those resolved. And as I showed you earlier, that no disease category actually decreased with changes that we made. So over time, our ability to tease out infants with some kind of immunodeficiency that's detectable at birth has improved. And the last category that I'll show you that I spoke about was the repeat pre-category. So this is data from 3,528 babies on the graph. And many babies that are born prematurely actually have normal TREC cutoffs, TREC values that are above the cutoff at birth. And so the majority of them actually do get released as screen negative at birth. But here we're looking at the cohort of babies who were born at less than 37 weeks and had low TREC values. And in those cases, as I mentioned, we do request a repeat specimen once the baby reaches 37 weeks of age, unless they have zeros right away. The vast majority of infants in this category resolve. But we are working with the Immune Deficiency Foundation and the Association of Public Health Laboratories on a study funded by the Health Resources and Services Administration to look at prematurity and factors that might influence the TREC values in this cohort. So maybe we can parse these kids out and maybe not request repeats from some kids if we can look at other demographic factors that might influence their outcomes. So our next steps are more of a study of T-cell lymphopenia using some molecular techniques and this participation in the SCID study that I talked about. Overall, I hope I showed you that, if nothing else, newborn screening certainly, for SCID, certainly evolved over time as knowledge increased and data accumulated. Population screening is all about very large numbers and ongoing data collection, and this is particularly important for rare conditions. So states inform each other of our findings and programs change over time to incorporate the increases in knowledge. We plan on continuing our study, and we hope that overall, the ultimate goal here is to only touch families who are truly at risk for some sort of immunodeficiencies and impact fewer over time unnecessarily. So with that, I'll thank you for your attention, and I'll pass it on to Colleen to talk about some of our work on the molecular analysis of our SCID positive infants. Thank you.
Can everybody see my slides okay?
Yes, Colleen, they look great. Thank you.
Okay, thanks. Thanks, Michelle, for starting us out. So we're going to change gears a little bit, and I'm going to share our experience with implementation of sequencing within the newborn screening program for SCID. So you should have a clear understanding now of our screening algorithm. So I'm going to drop down to those infants that are actually referred to a specialist for diagnostic testing. So our physicians at that point will typically order flow cytometry studies to look at the number of T cells and the different types of T cells that are present. They may order mitogen studies to look at functional analysis of those T cells. And then in some cases, they'll order molecular testing. Typically, it's a gene panel, genes involved in immunodeficiencies, But some of the problems that have been cited by our physicians is that the turnaround time to get the results can be very slow. They frequently encounter problems with insurance covering the molecular testing, and this all results in increased stress to the families. So we thought there might be some potential benefits to bringing the molecular testing into the newborn screening laboratory. We thought there was the potential to shorten the turnaround time to get results out, which would hopefully lead to a faster diagnosis, possibly be able to predict the type of immunodeficiency the child would have, possibly lead to early decision on the best treatment options, which would hopefully result in better outcomes. It would certainly be a cost savings to the families and hopefully result in less stress. So we were fortunate to enter into a cooperative agreement with APHL and CDC to look at the feasibility of doing the molecular testing within the newborn screening program. So there were several specific aims of the agreement. One is to develop and validate a second-tier multi-gene immunodeficiency panel using next-gen sequencing, to evaluate the utility of a targeted panel for SCID in a consented study. Would we be able to identify what the causative gene was? Would that in turn shorten time to diagnosis and lead to earlier targeted treatment? And then the third aim was that we would provide CDC with quality control reference samples so that when other newborn screenings wanted to bring on testing in their labs, the CDC could provide these reference materials to them for validating their own assays. The first step was to decide what genes we were going to include on our panel. So we looked at many of the commercial panels that were available and what genes were included in those. We looked at the CLSI guideline for SCID testing to look for suggestions for genes to include. And then we did a pretty extensive literature search. And we decided on this final list of 39 genes that we included in the panel. We selected the Illumina TruSeq custom amplicon reagents to target those 39 genes, and the assay was run on Illumina MySeq instrument. And then the Bioinformatics Pipeline for analysis of all the next-gen data was developed in-house. So although the Bioinformatics Pipeline is extremely complex, I kind of broke it down into 3 primary outputs from the analysis. So first is what's called a FASTQ file. And this is an output which converts the signal from the instrument into an actual DNA sequence. A BAM file or sequence alignment file is the output which aligns each individual DNA sequence to a published reference sequence. And then the variant call file is the output which identifies the differences between a sample's DNA sequence and that of the reference sequence. So this shows a typical sequence alignment of data obtained from next-gen sequencing. So each one of these DNA lines of DNA is a unique read of a patient's DNA sample. And it's, in this case, aligned to a reference sequence seen here. And in this particular case, this is a section of the ATM gene that's present on our panel. And the analysis software identified this particular location within the ATM gene where the sequence is different from what the reference sequence is. So you can see the reference sequence here has a G at this location, but you see a combination of reads that have A's and reads that have G's. So indicating this particular person has a heterozygous variant within the ATM gene. And our variant call file indicated this as the variant C-111G to A in the ATM gene. So once we identify all the differences in the sequences between the patient and the reference sequence, the next step is to classify each one of those variants for pathogenicity or the likelihood that it's going to cause disease. And for that, we use guidelines published by the American College of Medical Genetics and Genomics and the Association of Molecular Pathology. So these guidelines provide criteria for weighting different types of evidence for variant pathogenicity. And then it provides a set of rules for combining the different weighted evidence to arrive at a final classification. And there are 5 classifications for variants: pathogenic, likely pathogenic, variant of uncertain significance, likely benign or benign. And we report out those that are pathogenic, likely pathogenic, or uncertain. We do not report out the likely benign and benign, but that information is available to physicians if they'd like that information. So before we could offer the test clinically, we needed to validate the assay and submit it to our regulatory program. So to do that, we chose 25 samples for sequencing. 24 were from infants who had been referred for low TREX, who were diagnosed with either SCID or some other immunodeficiency syndrome. And we also included one reference sample. This is a cell line DNA that has published genotypes, so a published sequence that you can compare your results against. And this is a cell line DNA that many labs use for next-gen sequencing, either as a control or in their validation. And then for any variants that were identified by next-gen sequencing, we use Sanger sequencing, which is frequently considered a gold standard for identification of sequence variants, to confirm any results that we got on next-gen. So for 8 of the samples in infants diagnosed with SCID, we did complete Sanger sequencing for all 39 of the genes. And in the remaining samples, we only Sanger sequenced the pathogenic, likely pathogenic, or variants of uncertain significance that were identified by our next-gen sequencing assay. And then we also assessed the reproducibility of the assay through replicate testing, both within runs and between runs. So looking at the accuracy of our assay, first at the level of the reference sequence that we included, you can see here we identified all 76 of the known genetic differences in this particular sample from the reference genome. And we did not miss any of the true positives, giving us 100% sensitivity of the assay. We did, however, identify 3 false positive variants, which did not confirm by Sanger sequencing and were not present in the published genome for the control. So our specificity is just under 100%. We also use this control reference material in every run of next-gen sequencing that we do, and we require that in each run, this control give us 100% sensitivity. So we identify all of the variants in that sequence, and we have less than 10 of those false positives. The other accuracy that we're even more interested in is, are we actually able to identify pathogenic variants with our assay? So these are the 8 samples that had a diagnosis of SCID, where the molecular basis was known from testing in a diagnostic laboratory. And for all 8 of those samples, we identified the same genetic variant or variants that were determined in the diagnostic laboratory. So we submitted our validation to our clinical lab evaluation program, and the assay was approved to offer for clinical testing in July of 2018. So moving on to our consented study, our proposed algorithm would be that infants would be screened for TREX as they normally would be. Any infants that were referred to a specialist would have the same diagnostic testing that would routinely be done. If the physician determined that there was some type of immunodeficiency, they could obtain informed consent from the family, and we would test using our 39 gene panel. So of our 10 specialty care centers, 8 obtained IRB approval to consent their patients into the study. It was a 3-part consent: consent to SCID panel testing, consent to participation in an immunodeficiency registry for long-term follow-up, and then final was consent to provide a blood sample to the CDC for developing quality assurance materials. And participants can consent to 1, 2, or all 3 parts of the consent. So we consented 23 infants into the study. All 23 consented to having testing using the multi-gene panel. 11 consented to the long-term follow-up registry, and 2 families consented to obtaining a blood sample to be sent to the CDC to develop quality assurance materials. Of the 23 infants tested, these are the diagnoses. that were made, there's 1 case of classic SCID, 1 case of variant SCID, 2 were leaky SCID or Omen Syndrome, 6 were idiopathic T-cell lymphopenias, 4 had secondary immunodeficiencies or syndromes or some other immunodeficiency, and 9 actually had a final diagnosis of no disease. So although our suggested algorithm was that infants would have at least some initial diagnostic testing done, some of our physicians were very excited about our consented studies, so consented them at the initial visit before they had a confirmed diagnosis. So many of those infants ended up having no disease at the end. So looking at the variants that were identified in these consented individuals, first looking at the SCID group, we were able to identify a hemizygous likely pathogenic variant in IL-2RG in our case of classic SCID, and homozygous likely pathogenic variant in the RAG1 gene in 1 case of Omen Syndrome. We did identify heterozygous variants of uncertain significance in the case of variant SCID and the other case of leaky SCID Omen Syndrome. However, we did not identify a second variant in either of these cases, which brings up a limitation of our assay. Our assay was designed to only interrogate the coding sequences of each of the 39 genes and the splice sites. So we would miss any variants that were deep within intronic regions or in regulatory regions. And the assay is also not validated to identify any large deletions. So there is a possibility that these infants would have had a second variant in one of those genes. This is our idiopathic T-cell lymphopenia group. So there were 6 in this category. And as you can see, all but one of them had at least one heterozygous variant of uncertain significance. Some of them had multiple variants of uncertain significance. However, because they are of uncertain significance, we do not know whether these variants are actually contributing to the phenotype, to the lymphopenia that these infants have. Looking at the genes that are involved, several genes on our panel had variants identified. About the only thing that you might notice is that 3 of the 6 had variants in the FOXN1 gene. None of the variants were the same between those individuals. So again, we don't know whether these are just unrelated to the phenotype or if they may actually be playing some role. And another reason that we want to look further at this population molecularly. Looking at our other immunodeficiencies, either secondary T-cell lymphopenias or syndromes, we had 1 case of B-cell deficiency. Again, for 2 of the cases, we identified heterozygous variants of uncertain significance. No clinically significant variants were identified in the other 2 cases. So again, we do not know whether these variants played any role in the phenotype. And then although you might have expected that with the infants that had a final diagnosis of no disease, we wouldn't have found any variants, that was not the case. For 4 of the 9, we didn't, but for 5 of the 9 infants where the final diagnosis was no disease, we actually did identify heterozygous variants of uncertain significance. In 1 case, 3 different variants of uncertain significance. Just suggesting that we probably all have these variants of uncertain significance in our genomes that may or may not have any impact on disease. So how did we do with our turnaround time? Since that was one of the questions we were wondering in the beginning, we were able to report out results for our molecular testing in an average of 14 business days, ranging from 9 to 21. And looking at several commercial laboratories and their turnaround time is listed on their websites, we were at least as good as some of the laboratories and actually a faster turnaround than several of the other diagnostic laboratories. So we felt that it was reasonable for us to do the testing in-house and not delay molecular results at all. So in summary, we successfully validated and implemented a 39 gene SCID panel for clinical testing within the New York State Newborn Screening Laboratory. In our consented study, we sequenced 23 infants with low TREC values using our panel, and we're able to identify a homozygous likely pathogenic variant in IL-2RG in one case of classic SCID, and a homozygous likely pathogenic RAG1 variant in a case of Omen Syndrome. Our turnaround times were comparable to or faster than those listed by diagnostic labs, which should allow for prompt treatment, at least in our one case of IL-2RG SCID. That infant received a transplant at 3 months of age and was doing well at our last update. And in several cases, molecular testing was also done at a diagnostic laboratory at the same time that we were doing the testing in-house. And in those cases, when testing was also done in a diagnostic lab, our results were always concordant with what the diagnostic lab reported, which is very reassuring to us, as I'm sure it was also to our clinicians. Enrollment in our consented pilot was somewhat lower than we were expecting, although when we talked to the clinicians about it, many of them cited difficulty in identifying an appropriate time to approach parents when they were already overwhelmed by the diagnosis to begin with. So it was trying to find the time to approach families to ask if they wanted to consent to the study. And then probably the most negative thing is that most of our variants identified in the study were of uncertain significance. So we just don't have enough knowledge yet about the genetics of these immunodeficiencies to classify the variants. And we're hoping that with further molecular studies in infants diagnosed with immunodeficiencies, it will improve our ability to classify these genetic variants and enhance our understanding of how they're contributing to disease. So where are we going next? Very shortly after we designed the 39 gene panel, we were informed by Illumina that they were discontinuing the TrueSeq custom amplicon reagents that we had used to design our 39 gene panel. And so we knew we were going to have to redesign the panel at some point using a different technology. So we've now done that. We've redesigned the panel using ArcherDX VariantPlex custom reagents. And since we were redesigning, we decided to review the literature again, and we've now expanded our panel to 55 genes. So we've added these genes on the right, and we've also targeted some common deletions that have been shown to be associated with immunodeficiencies in a few of the genes. And this new panel is currently being validated. And once validated and approved by our clinical lab program, the first study that we'll undertake is looking at those idiopathic T-cell lymphopenias and doing a molecular analysis in that population. So I just want to acknowledge the sources of funding for the various SCID testing that goes on within our newborn screening program, as well as acknowledge the members of our New York State newborn screening program involved in SCID screening and in the consented study, as well as our New York State SCID specialty care centers and our Wadsworth Center Applied Genomic Technologies cluster, which is our sequencing core facility here within the Wadsworth Center. Thank you very much for your time, and I think we have some time for Michele and I to answer questions.
Thank you so much, Colleen and Michele. The chat is open, so if anyone has any questions or comments, I already see there have been a few. Feel free to type them in there. I will also open the all participants' ability to unmute themselves if you have a complex question that might just be easier asked verbally than typing it out. So you should all be able to unmute yourself if you have something to share. Thank you again, Colleen and Michele.
Okay, so this is me. Can you? Okay, I'm going to ask one for Michele and Colleen. Wonderful work. I'm wondering, did you have a plan to change this consent model to our more standard care? And I think it's wonderful when, especially if you really limit it, with education, everything is when you only open to the patients with flow has done indicated true lymphopenias or whatever. So potentially not have the consent model anymore. Any plan on that?
You want me to get it?
Yeah, I mean, Colleen and I are kind of looking at each other. We talked about different versions of this. Colleen,
Right.
do you have any? I mean, when do you do it? Do you do it? Funding, that sort of thing.
Right. So obviously funding is an issue. How many kids can we afford to screen? And I think without at least some indication that there's immunodeficiency, we will end up with variants of uncertain significance that we don't really know how to interpret anyway. Now that we have the assay, or we'll have the new panel approved by our clinical lab program, we can offer the test with a simple genetic testing consent, as opposed to our pilot study that was a much more involved consent form. So that might be less burdensome for physicians to do consent as well.
One more quick, I thought. So when the physician sent back a request for the sequencing, did you guys require some kind of like flow testing result, function analysis result? Did you ask for that?
We did not. Yeah.
Yes, because it was a multi-part study, and part of this was trying to figure out when was the best time to actually even get consent. So like Colleen said, some of them were kind of gung-ho and enrolled people early, which was good, but they ended up, not having a DX. So you have to think about what would be most beneficial for the family and for the, for the field as well.
Thank you.
Hi. So if you were thinking of expanding this, were you considering like choosing, a CD3 cutoff to select kids, like under 300 or under 500 or whatever, to avoid so many VOUS? That's my question.
I mean, I don't think we haven't really considered it because we haven't really gotten that far, I guess. Again, we were trying to sort of figure out what model would work best and be able to provide information. So part of the idea was that you had some kids who kind of went through more of a diagnostic odyssey. And if we provided that, do we provide that information on any baby with 0 TREX, or 125 or less, or when they're seen regardless of the referral? So we kind of had a lot of things to think. Still have a lot of things to think through about what would be most useful. And again, we want to avoid, even if you use that cutoff, I think we're still going to find variants that we don't know how to interpret and confuse the situation more than a phenotype. I don't know if you have anything else.
I'm finding our physicians in California have been doing a lot of genetic testing. They do order it and get results, and they never report the VOUSs to us, so we don't know. But we see a lot that you have mentioned.
I think there's
and VOUSs are a problem. So yeah, sorry.
Yeah, there are some labs I think that choose not to report those, right, Colleen?
Yeah, that's correct. Some will only report pathogenic, likely pathogenic.
Thank you.
Colleen and Michelle, I don't know if you've seen the questions or comments that have come in on the chat, someone is wondering where the 2 patients found as leaky omens actually found to have omens, or was their final diagnosis RAG1 and IL-7?
So I believe the final diagnosis on the RAG1 that we identified was Omen Syndrome. It was an older Amish family, and that was the diagnosis we were given by the physician.
Thank you. I see another question coming in. Sorry. I see another question coming in about the merits of CREC testing or CREC versus TREC testing.
Yeah, so we had looked at CREC testing and we had entertained it, I think, at some point earlier on, and we've gotten kind of conflicting feedback from our providers about whether that's actually useful or not. I see Carlos is on the line. He was in kind of oversaw a lot of that study, if he wants to chime in about the utility of CREC. I don't know if he wants to chime in or not. I mean, you can pick up combinations of things with CRECs and TRECs or CRECs alone, you know, and make determinations. Some people commented on the rarity of those conditions and whether we would catch them or miss them and that sort of thing. So that was sort of left in a holding pattern right now, at least for us.
Someone is also asking if you're confirming variance by Sanger before reporting.
Yep.
Wait, can you hear me now?
Yes.
Okay. Yes, we Sanger confirm everything.
And then I see another question asking, how are the immunology and genetics clinical communities collaborating in New York? What should or could states do if thinking about moving in this direction? How to support families and clinicians?
I mean, we work closely with our specialty care centers. We have 11 of them in the state. They all are, some of them do order genetics and a lot of them have genetics at their centers as well. So I'm not quite sure what more specifics maybe on the question. I don't quite, or if Joan wants to speak up and kind of clarify for us what she's looking for.
Yep. Yeah, I figured out unmuting. So, so my question was basically about access to genetic counseling for families, and I was just curious if there's a kind of a recommended model, standard model, or if it's basically up to the centers and how they work to help families understand the diagnoses.
So the way our specialty care centers are set up in our state is that they're in what they call health service areas. So they're located across the state based on where the family lives. And we have genetic centers. I'm pretty sure, I'm trying to think on the fly here, I'm pretty sure they all have genetics associated with them. And I know some of our specialty care centers utilize genetics at their centers as well. I don't know how well coordinated it is overall, but we certainly have a group of geneticists that are across the state with counselors that help in the metabolic diseases, CF, and basically they can see any of the families that get referred.
Thank you.
Yeah, and Carlos commented that our physician were a little ambiguous about adding CRECs to the TREC assay. And then, that would have to be done on top of the SMA and the TREC assay as well at this point, so.
Well, thank you so much, Michele and Colleen. I don't see any other questions coming in, but we can stay on the line for another minute or so in case someone has a last minute thought or something they would like to share. But Thank you all for joining us today on the first day of the 2021 SCID Compass Summit, and a huge thank you to Michele, Colleen, and all the other providers and experts who have presented a session today. If you couldn't partake in anything that has happened yet, everything was recorded, and if you just go back to the session, you should be able to watch the recording and navigate to any of the resources that we have on our website and through our program. So thank you all so much. We hope to see you again tomorrow, again at 11 a.m. Eastern. We have a presentation on SCID variants in the Irish traveler community in the United States, as well as a session on special considerations for screening premature infants, as well as a Spanish session. So lots of exciting things to come tomorrow. And thank you all for taking time out of your very, very busy, hectic days to join us here on Zoom. Thank you again, and take care.
Thank you.